Good morning, everyone. Welcome to CCMB Biologue. We have with us today Dr. Vatsala Thirumalai. Vatsala completed her BTech from Anna University. She moved to Brothers University to pursue a PhD in neuroscience. She had postdoctoral stints at CSHL and NIH. Currently, she is a group leader at Neural Circuits and Developmental Biology Lab at NCBL. Welcome to CCMB Biologue, Dr. Vatsala. We are looking forward to follow the beat.
Thank you so much, Saujanya, and the entire team of CCMB Biologue, including Kaushik, who had written to me. Thank you so much for your interest in our work. As I was saying earlier, I would love to have come in person and given this talk, but maybe conditions permitting in the future, we can always do that. But today I'm very excited to share our latest science with all of you. Please feel free to stop me in the middle, interrupt me and ask me questions in case I'm not clear over any concept. So without any further ado, let's go ahead. And I hope all of you can hear me clearly and also see my slides.
Yes.
Yes? Yeah. Okay, fine. So what I'm going to be talking about today is about how our brain perceives time intervals between sensory events, things happening in the environment, elapsed time, so to speak, and how it uses these time intervals for use for behavioral adaptations. So why is time discrimination important? So time discrimination happens in animals. Animals can discriminate time intervals over a very large range from 10 to the -6 seconds to several hours. For example, if you take the microsecond range, time intervals in the microsecond range helps us localize sound. So for example, you're sitting here and you hear a noise on your right-hand side, the sound waves reach your right ear earlier than they reach your left ear. And this delta t is usually not very long. It's in the microseconds range. But yet we are able to localize the sound because we have neural mechanisms for distinguishing even such very, very small time intervals. Similarly, there are neural mechanisms on the other end of the spectrum over the several hours range, which is the circadian rhythm that I'm talking about, where animals can, process elapsed time over 24-hour cycles, even in complete darkness. So if you put an animal in complete darkness, they can still follow the circadian rhythm with a certain little bit of jitter, but more or less they are able to follow it because there are circuits in the brain and elsewhere in the body where gene expression is controlled over these time scales. But in the middle range, which is in the milliseconds to the several seconds and minutes range, we don't understand the brain mechanisms involved so clearly. But this range, which is from the milliseconds to seconds to minutes, is the behaviorally more important one, because this is where conscious perception of time happens. So, for example, much of music and speech, where when I'm talking, I'm leaving spaces between words, and that helps you parse the words and understand the meanings. And also there are time intervals between syllables. I stress on certain syllables more than I do on others. So these happen in the millisecond time range. Over the seconds, minutes, and few hours time scale, behaviors can be adapted. So this example that I've shown you here is about a hummingbird, where the study that appeared in Current Biology in 2006 showed that hummingbirds can learn the time intervals in the several minutes range, so 10 minutes and 20 minutes. What they did was they placed an set of artificial flowers. Some of them were refilled with nectar every 20 minutes, and some of them were refilled with nectar every 10 minutes. And the hummingbirds could learn which flowers were refilled at 20 minutes and which flowers were refilled at 10 minutes, and they revisited these flowers at the appropriate time interval. So you can see that they visited the orange flowers roughly around 20 minutes only, and they visited the blue flowers roughly around 10 minutes, and these blue flowers are the ones that were refilled every 10 minutes and vice versa. So this shows that animals can process time and learn the elapsed time and know when to expect what will happen at the appropriate time. And this also applies to humans. For example, when we are sitting at a signal light, many signal lights these days have a time ticker next to it, but many signals also don't have it. But suppose you reach a signal and it has just turned red, you know that you can't expect it to turn to green immediately. So then you know how much time you need to wait before you start expecting the light to change. So behaviors, and you get ready for starting your vehicle once the light changes. So behavioral output is modulated by our ability to discriminate time. But the problem is that we don't understand the neural mechanisms underlying how time intervals are processed in this broad range of several hundred milliseconds to seconds, minutes, and hours. And so we decided to look at this question. And one of the regions that we wanted to focus on was the cerebellum, because the cerebellum is known to be important for temporal coding and also be involved in motor actions. So this work was primarily driven by a graduate student in the lab, Sriram Narayanan. He imagined the project. He put together the machinery needed to collect the data. He collected the data and he analyzed. So he's a one-man army by himself. I'm just talking about the work. He's the prime driver of this work. Alok Verma did the electrophysiology experiments which support the results that came out of Sriram's work. And Jagadish maintained the fish facility in an excellent condition that allowed us to do this work. And this work is now also on bioRxiv. So if you have any further questions, you can always go back and look at this paper for further details. So coming back to this question, the cerebellum is an important motor organ, and it has been shown before to be involved in timing in the sub-second range. So we wanted to look at whether it was also involved in longer time range learning and whether that can be applied to motor actions. So we used zebrafish larvae for these experiments because these larvae are transparent and the brain can be imaged while they're doing interesting behaviors. And also they have interesting motor behaviors, and one such behavior is the optomotor behavior. It helps the fish larvae stabilize themselves in moving water. As you know, these fish are freshwater fish. They live in shallow streams and the water is moving, it's slowly moving. And if they perceive that the current is washing them away, then they will swim to compensate for the current and then try to stay in one place. And this response is called optomotor behavior. You can recreate this in the lab by showing black and white bars known as gratings. And in this video, you will see that initially the larvae are uniformly distributed over this tub. I hope you can see that. And the grating will move from the right-hand side to the left-hand side. And you will see that at the end of the video, many of the larvae have come to this half of the tank. Let's see the video. So here they are swimming around spontaneously. Now the grating will appear. And let's see what happens to the localization of the larvae. Now, you'll see that the grating is flowing. And let's see at the end of the video where the larvae are located. And you see that most of them have now migrated to this half of the tank. So this is optomotor behavior. And obviously we can't do this in freely swimming fish because we need to hold them in place to image the activity of the brain. So we can recreate it by head fixing the larvae, where the head is stuck in a jelly-like material called agarose, which many of you are familiar with. And the tail is free to move. And then we show the gratings underneath, and it evokes tail movements, left and right tail movements indicating forward swims. So this is what we do. We embed the fish in agarose and we put it under a microscope and we look at brain activity. So how do we look at brain activity? Sriram made a custom 2-photon microscope, and we put genetically encoded calcium sensors into Purkinje neurons of the cerebellum in the brain. And underneath the fish, we put a screen to show the gratings, and a little piece of the screen was cut out so that the tail can be visible. And we did something called closed loop feedback, where the tail movement was used to detect swims, and then the swim was used then to move the screen back. This is to give realistic feedback to the fish, so it continues to respond during the paradigm. Otherwise, it gives up and doesn't respond. So this is what that looks like. So this is the stimulus, and you can see that when the tail is moving, then the grating is moved in the opposite direction, and more or less the position of the bar is maintained, right? So that's the closed loop feedback. And then simultaneously, we are imaging the activity of Purkinje neurons expressing GCAMP, which is a genetically encoded calcium sensor. Whenever the neuron is active, then calcium flows into the cells, calcium levels are elevated, and that causes a transient increase in fluorescence, which we can measure using our custom 2-photon microscope. And the stimulus that we give, again, is not a very constant grating stimulus, because our original question is to look at time intervals, right? So we repeat the stimulus every 5 seconds. So what we do is there is a stationary grating, which is shown by the 0 millimeter per second flat line here. And then it switches to 10 millimeters per second grating flow for 2 seconds, and then comes back down to no flow. So stationary grating for 5 seconds and moving grating for 2 seconds. And during this 2 seconds, they also receive the closed loop feedback. So the fish is repeatedly presented this repetitive optical stimulus every 5 seconds for about 200 times. And then what we do is that in about 10% of the trials, randomly, we don't give the stimulus where it should have occurred. So here, the optic flow should have turned on, but it doesn't. Okay, so this is a probe trial. So this is very important. I'm going to be talking about flow trials and probe trials. So it's very important that you understand this, that during flow trials, the optic flow is on and the gratings are moving. And during probe trials, the optic flow should have come on at time t, but it doesn't come on, and the grating stimulus is stationary. Okay, so what do we see? So this is what our data collection looks like. We have a camera that is looking at the tail movement, and we have the imaging set up to look at the brain activity where neurons are flashing on and off. And then offline, we can look at the fluorescence change in each of these cells during the entire experiment. And you get roughly around 60 to 70 neurons in one plane. And you can get the fluorescence intensity profiles of all these 60, 70 neurons while the fish is performing these behaviors. So in sum, then from this experiment, we have the sensory stimulus channel where we have the optic flow with flow trials shown by these tall bars here, and probe trials shown by these long gaps that you see occurring randomly between trials. We have the swim velocity calculated from the camera following the tail beat. And we have the DF over F of Purkinje neurons. One neuron is shown here, but we have several neurons from the same fish. And then we can repeat this over many fish. And what do we see? So the way we analyze these data is that we calculate event-triggered averages. The event can be either the onset of flow or the onset of probe. And then what we do is that we say, okay, at this time, let's say the flow turned on. What did this neuron do? And similarly, we can do that for the multiple trials. And then we collect all of these calcium transients and we average them, right? So you can get for a single cell, what is the average flow response like? Similarly, you can calculate for a single cell, what is the average probe response like? Conversely, what you can do is, I told you that there are 60, 70 neurons. So let's say this is trial number 1. For trial number 1, this neuron responded like this. There'll be other neurons that respond differently. You can calculate a trial-wise average and you can say, okay, for this trial, the cells responded like this, and then you can get an average trial response as well. So what we found were principally two classes of cells, flow responding and probe responding. What do they look like? So here is an example. So bottom here, the ball shows you how the trial is progressing. And you can see that there are two colors that I've indicated here, blue and cyan. I'll play the video again. But if you look in the blue squares on top, those are the same cells whose DF over F is plotted below. And you'll see that these blue cells show a response when the flow is on. That is the top of the square wave shown below in yellow. Okay, let's see that. So they become brighter whenever the flow turns on. The cyan colored cells don't show much of a flow response. You see that their responses are dimmer during the flow trials. But this red line here indicates the time of the probe trial. And during the probe trial, these cyan cells will show a large increase in fluorescence. So let's see that again one more time. So you have principally these two kinds of responses, flow responses and probe responses. Now we can use these responses to, and this is on the left-hand side is data from all the fish, about 27 fish. The flow response profiles of all the neurons in all the fish, about 1800 neurons in all, and the probe response of these cells. So we can use hierarchical clustering to then come up with classes of neurons among these Purkinje neurons. And that's what we did. So we did correlation-based clustering. So principally, there are three kinds of probe responses: small probe response, large probe response, and absent probe response. And similarly, small flow response, large flow response, and absent or negative flow response. So you can imagine that there are 9 different combinations of these 3 response profiles of the flow responding and the probe responding neurons. But some of these classes are not well represented. There are only 1 or few cells per fish. So those are the 4 corners of this square. The middle 5 classes are what I'm going to be talking about. And among these 5 classes are 3 classes, which is the vertical bar here, which have very large probe responses. And the 4th class, which has modest probe response. And then finally, a 5th class, which has a very large flow response, which is this orange color here, but no probe response, right? So these are purely, cells that report that, okay, flow happened, and it doesn't report whether a probe trial happened. That is, flow onset was expected, but did not happen. That job is done by 1, 2, 3, and 4. These classes of cells are the ones that report an error. So that is what I'm going to show here. So these 4 classes are the ones that show that an onset of a grating flow was expected, but it did not occur. This cluster over here shown in gray is a cluster which tells you that, okay, I expected a flow and it happened, right? So there are these 2 different classes of neurons, even within the Purkinje neuron class in the cerebellum. So the question then is, what do they do, right? I will leave out some of the details and I'll stop here to take any questions that may be there. Are there any questions this far?
I don't see any raised hands, but if I may ask, what happens when there are, say, multiple stimuli?
No, we haven't tried that. That's a very good question. When you combine, let's say, I suppose you're talking about multiple modalities, right? Visual, sound, and so on. Is that what you're talking about?
Yes.
Yeah, that would be fantastic to try. But the problem is that then you have to do a very controlled experiment where first, the parameters of the stimulus will have to be tuned and the combinations of the parameters will have to be tuned. So you're looking at, a large parameter space. And so we've not had the courage to attack that problem yet. And so we stuck to one modality only in these experiments, but that's something that can be done in the future. Once we understand the visual sensory processing much better, then we can move on to auditory and combine these responses and ask whether there are multimodal neurons as well. Okay, so I will move on. So the question is, okay, I told you that I'm giving the fish these repeating sensory stimuli, and these repeating sensory stimuli are making the fish learn the time interval between the stimuli, and then that leads to the fish expecting the stimulus to occur at a certain point of time. And therefore, if the stimulus does not appear at that time, it reports an error. That is what these probe responses are. Interestingly, these probe responses develop very, very quickly. So when we presented a probe trial in the third trial itself, then we saw a response which is shown in the dark blue color, indigo color here, which is equal in amplitude and probability to responses that we may see later on in the experiment, let's say in the 12th position, where it might have experienced the periodicity of the stimulus many more times. So what this tells us is that the fish is able to look at the time intervals and learn it very, very quickly. And that data is summarized on the right-hand side. We won't go into the details. But what this tells you is that this is a mechanism that can happen fast. It doesn't require a very long-term training for the fish. Okay, I'll tell you how we think it is happening. Your question might be that how is it that a fish is able to learn the time intervals within 2 trials? We'll come to the mechanism in a little bit. The second interesting feature of this probe response is that it is time locked to the expected onset. So the red line here is the expected onset of the flow trial, and it did not happen. And the response is time locked to it. And you can see that, if you change the time interval and retrain the fish to a different time interval, then that response also moves. That's what we did here. So initially we trained the fish for 5 seconds, intertrial interval. Then we moved the intertrial interval suddenly to 7 seconds. And within 2 to 4 trials of presenting the new 7-second interval, the probe response also shifted to this new time interval. So this tells you that it develops fast and it also adapts to new time intervals quickly. Okay, so Sudeepta is asking a very interesting question. Do the probe response cells learn from the rhythm of flow response cells? If so, how many minimum flow trials are required? Very good. So this is exactly what I'm going to tell you, that in fact, it depends on the flow responding cells and the information that they acquire. We'll look at it later on in the presentation. As above, for the minimum flow trials, the minimum that we've tested is 2 trials, and the 2 trials and by the third trial, if the probe trial occurs on the 3rd position, then you can get a robust prediction error response. And Banu is asking, do zebrafish have neuromast cells? And if they do, wouldn't the absence of actual current cause an error in stimulus prediction? Very good question. So they do have neuromast cells, and they are sensitive to, they exhibit rheotaxis. But vision is a dominant sensory stimulus, and our lab and multiple labs around the world have shown that just showing the visual stimulus is sufficient to trigger this response. And it's true for many animals that, they depend on vision to help orient them in their environment. And if the visual input is there, then it kind of dominates over all other modalities. But if you were to do it in the darkness, I would expect that giving flow stimuli would also trigger similar error responses that we see. That's a good question. Okay, so coming back, the next point was, okay, it develops quickly, but if you were to give it no other stimulus and that just let it sit, how long will it remember this periodicity for? And that's what we did here. We gave the fish a series of repeating flow stimuli, and then we stopped. So this is multiple probe trials occurring in quick succession. And you see that here are the flow cells, and the flow cells are producing very nice responses as the flow is occurring. And when the flow is stopped, they don't respond. There's nothing there, right? Conversely, you see these probe cells, and the probe cells have very small flow responses. And then when the probe trials occur, they start to produce responses. And to our surprise, even after 10 trials, the responses did not die down. They didn't decrease significantly in either probability or amplitude. And that was a big surprise to us. This, you can imagine this is about 70 seconds and the memory has still not died down. We don't know how much longer it lasts, and that will be an interesting experiment to try out. Okay, so I've told you that there are these two classes of cells, flow cells and probe cells. The flow cells are time-logged to the onset of actual sensory stimulus, and the probe cells are time-logged to expected flow stimulus, which does not occur. So it's sort of like a disappointment signal. I expected that the flow should be there, but it's not there. We wanted to find out what actually contributes electrically to these signals. Now, calcium imaging is useful for monitoring activity of populations of neurons, but it's not very helpful in understanding the exact ionic mechanisms that lead to activity, because calcium sensors are very slow, and a number of different processes can lead to calcium signals within neurons, especially in Purkinje neurons, which have two kinds of activity: sodium-dependent simple spike activity and calcium-dependent climbing fiber synaptic input. So here is a rough circuit of the cerebellum. So here are Purkinje neurons, and they receive two principal kinds of synaptic inputs. One from the granule cells via parallel fibers, and one from a nucleus in the hindbrain called the inferior olive via climbing fibers. Now, both of these are excitatory, means they depolarize the neuron and make it fire. And both are mediated by glutamate. But the parallel fibers inputs are weak, and there are many. And the climbing fiber inputs, in the larvae, there are about, there are not as many as parallel fiber inputs. There are a few, less than 10, I would say. And in the adult vertebrates, all the way from fish to mammals, there is only a single climbing fiber that innervates the Purkinje neurons. And these climbing fiber synaptic inputs are very strong as opposed to the parallel fiber inputs. So typically, these parallel fiber synaptic inputs are only about few tens of picoamps, while the climbing fiber inputs are in the hundreds of picoamps range. So both of those inputs are arriving into the Purkinje neurons. The climbing fiber input causes calcium entry into the dendrite because it is so strong, whereas the parallel fiber input can increase the sodium spike firing rate. Excuse me. So we asked, when you see these flow responses and probe responses, what exactly is causing these responses? What responses are being modulated? So we repeated the exact same experiment, but this time we paralyzed the fish larva. Excuse me. And we presented the same closed loop grating stimulus, but this time we are monitoring the swim output through motor recordings, electrical recordings. And we are monitoring the Purkinje cell activity, one cell at a time this time, using extracellular loose patch recordings. Okay. And we can distinguish through these loose patch recordings the simple spikes and the climbing fibers because they have different shapes and amplitudes. Okay, and what do we see? So I'm not going to bore you with all the details. Primarily, what we found was that the flow responses are mediated by both simple spikes as well as climbing fibers, while the probe responses are due to climbing fiber inputs alone. The simple spike rate is not modulated significantly during these probe responses. And that tells you that the error information is being supplied to the Purkinje neuron from the inferior olive via these climbing fibers. The flow responses are being supplied by both the inferior olive as well as the granule cells. And what does the Purkinje neuron do with these two streams of information? We'll get to that next. But to summarize, using imaging, we found two distinct subgroups of Purkinje cells. One cluster which ramps up activity prior to flow onset. I sort of glossed over that. We saw that both in the electrophysiology as well as in the imaging, where the activity of these flow responding cells climbs up a ramp even before the onset of the visual stimulus. Fine. Why this is important, we'll see in a little bit. And there are other clusters which are error responding cells, which say that, okay, I expected flow to occur, but it did not occur at this expected time. So the time interval is very, very important. It's time locked to that expected flow time. And these error responses develop quickly, they are long lasting, and they are adaptable to different time intervals. And the PE signals are fed into the Purkinje cells by means of PE means prediction error signal, the probe signal, are fed into the Purkinje cells by the inferior olive. Fine. So why is this useful? Why should we even care? We should care because it modulates the behavioral response. One feature that, so let's step back a little bit. What might the fish be doing with this information? So you present visual stimulus that is repetitive, and the fish learns the time interval, right? It learns that, once a flow is stopped, it knows that the next flow is going to happen in a certain amount of time. What might the fish be using that response, that information for? The latency, which is the time from the onset of the stimulus to initiating the swim. It needs to swim in response to flow, and that's how it maintains its position, right? So if the fish is able to quickly generate a flow response, quickly generate a swim, then it's behaviorally salient for the fish. It helps it maintain its position in the water body, in freely swimming fish. So we asked whether, this kind of predictions and prediction errors are used by the fish to modulate latency. And the way we looked at it was that in the same fish, we looked at different trials. There were some trials where many Purkinje neurons generated an error response. And there were some trials where only a few Purkinje neurons generated prediction error responses. So that's the probability P value here. P equals 0.26 means 26% of the probe responding cells actually produced a significant probe response. Now, during a probe response, there's no swimming. So we looked at the swim latency near these such high probability event probe trials. And we asked what the latency was like. And we found that the latency was negatively correlated with the prevalence of these probe responses. In other words, when the probability of finding an error response is high, then as soon as a flow comes, the fish is generating a swim quickly. That means it has a very good idea of what the interval is like at that time point. Conversely, during different trials, when the confidence in the time interval is low, then the error responses are also less probable, and the latency to generate swims is also longer. That's what is shown in this graph over here, that if you have a good confidence of the time interval, then if it doesn't happen at that time interval, you produce robust error responses. And when the flow happens at that time interval, you produce swims that also happen quickly, right? That makes sense. Now, is there more evidence for this line of thinking? Yes. In the flow responding cells, the activity that ramps up prior to the flow onset, that also negatively correlates with the latency. Okay, this is important because the flow responding cells are not actually driving any motor action. It is, sorry, the probe responding cells are not driving any motor action. It is only the flow cells that are driving the motor action. So they have to have information about the latency of the upcoming swims, and indeed they are negatively correlated, which means that, again, greater the confidence in the learned time interval, greater the activity before the onset of the flow, and lower the latency to produce swims. So how do we test this? So what I'm telling you is that this is more like a trial to trial modulation of swim latency. It is not a long-term modulation of swim latency, which also happens, by the way, but that's a different process. How can we test that there is a trial to trial modulation of latency? Again, we got a handle on this because we know for sure that when a probe trial happens, the trial immediately following it, the latency is a little bit longer. Okay, so here is the probe trial, and here is the trial that follows immediately after it, and the latency is a little bit longer, significantly longer compared to the baseline latency level, and then it quickly comes back. So we asked whether the cerebellum is indeed involved in this trial to trial modulation by lesioning the cerebellum using the laser. So we lesioned off the cerebellum, and we find that the trial to trial modulation of swim latency is gone. The fish is no longer able to use history to compute future, right? So that was indeed the evidence that, in fact, these cells and what they encode, the predictions and the prediction errors, are useful for directing behavior. So in summary, what I've shown you is that there are two distinct subcircuits that drive interval timed behaviors. There's one class of Purkinje cells, which are the flow cells, which are active only during flow trials, and they are responsible for behavior generation. They direct the latency of the swim in that particular trial. And there are other classes, which are the probe responsive cells, which learn the stimulus timing from the activity of the flow responding cells, and they use that information to update when the actual time intervals are no longer matching with reality, because you can't be stuck with one set of time intervals. You need to constantly adapt, and the adaptation is done by the probe responding cells. So that's our model, that there are in fact these two clusters. The swim decision is being directed by these flow responding, cluster 4 is the flow responding cells, and they drive the swim decisions, whereas the probe responding cells are used to update the learned models and keep them up to date, so that when things change in the environment, you're not stuck with an earlier version of what it used to be. So I will stop there and thank all the funders for helping us, and the facilities at NCBS that allowed us to do this work. And that is Sriram, who is the primary driver of this project, Alok, who did the electrophysiology, and Jagdish. Thank you very much for your attention. I hope I didn't, overwhelm people with a lot of technical details.
Thank you, Vatsala. That was a very beautiful setup to understand, to follow the beat. I don't see any questions on Zoom, but we have a technical question that was posted on Facebook where Santosh wants to know how you fix the larvae without anesthesia to record the optomotor response under the microscope.
Right. So we anesthetize them while embedding them. I suppose he's asking about the imaging experiments. So we anesthetize them with trichine, and then we put them in low melting agros. And then once the agro settles, then they are stuck in place. And after the experiment is over, we can always release them and they are fine. And they, in fact, that's a test that we do to make sure that the neurons are healthy. That after the experiment, the fish are capable of swimming around and being happy.
I don't see any raised hands or questions in the Q&A box. Sudipto has a question.
Yeah. Hi, Vatsala. It's an amazing talk.
Thank you.
Can you hear me?
Yeah, I can hear you.
Yeah. So, there's a lot of questions I have. Like, I'm not from this field. So this is very like intriguing talk. So so you have found out that there are like different set of cells which are taking cues from the environment, and some sets are behaving differently, like to like learning from the others. So what do you think, like what are the like difference molecularly, like they have different set of receptors or something which helps them to learn things and then keep as a memory to store it, like how it works mechanistically, like
so
thoughts?
Yeah, that's a deep question. We don't know, but I can speculate. So the memory, I in fact, thanks for bringing it up. So the way we think this happens, because it does happen over 2 trials, is that the brain is capable of generating endogenous rhythms, and when an external stimulus is presented with a frequency that is close enough to the brain rhythm, the brain rhythm can get entrained to the externally applied frequency, right? So that is how we think this is happening, that there are these cells either within the cerebellum itself or in the other centers that are talking to it, that have an oscillation going on of a certain frequency. And then when we apply these external stimuli, that rhythm gets entrained to this frequency. When we change the frequency, then it can get entrained to a different frequency. So of course, that means that there is a certain range over which the entrainment can happen. It cannot happen over a very big range, and there may be different oscillations that track rhythms of different frequencies. And that's how we think this memory concept is being implemented in the brain, by means of entrainment to externally applied rhythms. I hope that answers your questions.
Thank you. Yeah.
We have a question from Ajay. Does the speed of the panel 2 affect the response of the cells, the black and white grid that's moving?
Absolutely, absolutely. So zebrafish can follow speeds from about 1 centimeter per second to about 5 centimeters per second, and their responses, they swim with a certain speed which matches the speed of the grating. So of course these responses will also be modulated by the speed of the grating.
I have a question. In the same setup, say we change the stimuli and we again measure the Purkinje cells firing. Do you think the same subset of cells will have a similar response, or say the cells that had delayed firing in one stimulus will have a faster response?
Can you repeat that question? Sorry.
We have a stimulus and there are subsets of Purkinje neurons which fire at a given point of time. And we have a different stimulus now. Will these subsets remain the same, or say now there are another subset which fires first and then later?
No, so at least for the when we showed the 5-second to 7-second intertrial interval, we selected cells that were active for 5 seconds, and we asked whether there are cells, these 5-second cells now respond to 7 seconds, and a significant proportion of them did. Of course, there are other cells which can also respond, but predominantly the same cells can switch from one rhythm to another. Is that your question?
Yes, mostly because the audiovisual perception in real life is much more complicated. At a given point of time, we have too many patterns to recognize, both audio and visual. So it's difficult to keep track of each and distinguish each of them.
Correct. But so these cells are very removed from the primary sensory cells and the primary sensory areas. For example, if you take the optic flow stimulus, the retina processes the visual information, and then in the fish, the primary visual areas are in the optic tectum, and from the optic tectum, then the information comes to cerebellum. So already it has crossed multiple synapses before it gets to the cerebellum. So it's not a primary sensory response. It's actually a very filtered version of the sensory response. Okay, so it is going back to your earlier question of the multimodality. These cells are capable of responding to multiple features of the stimulus field and also to multiple stimulus modalities. So it's possible for them to switch from one frequency to another, and also from one speed of the grating to another.
Okay. Thank you. And we have a question from Bhanu Pranav. Since these are larvae and the functional connections would be different in an adult, what do you think this would imply for the rhythms and the error prediction in adults?
Yeah, very good question. Although these are larvae, the larvae are independent organisms and their motor behaviors are quite mature. So the optomotor response is also mature and similar to what you might see in the adults. So if you see such mechanisms in the larva, I would assume that the adult also uses similar mechanisms to process optomotor responses, time intervals, and so on. But that is something that needs to be checked out in the adult animal as well. But can't do it in zebrafish because the adult zebrafish is not transparent. So then you'll have to do invasive surgery, and you don't have access to as many neurons. So all of the problems that you face in, for example, mice, you will face in the adult zebrafish, and it fails to be an attractive model organism for questions such as this. So your best bet is to look at behaviors that are quite mature in the larva, and then use the transparency of the larva to address questions in this neural basis of behaviors.
I don't think we have any more questions. So
okay,
I would like to thank you for enlightening us about the neural circuits. And we would love to host you in person in CCMB sometime soon.
Thank you, Saujanya, and thank you to all the attendees for coming in and for your questions. It was a pleasure to give this talk.
Bye.
Bye.
Take care.
You too.